Good morning, everyone. I'm Loïck Moissonnier, a PhD student in drug resistance and one protein group in Lyon. So today I show you how I use time-resolved approaches to understand the mechanism of an ABC transporter. So the context of my talk is about antibiotic resistance. According to World Health Organization, it counts 700,000 deaths per year and it will increase in the future. Therefore, it's a major health concern in a society. So, in fact, there are many molecular causes of resistance, like the inactivation of antibiotic, the modification of the target or protection, and so on. But but my talk focuses on proteins in charge of active efflux of drugs, lower this concentration inside of cell. And I work on one of them. It's BMRA, it's an ABC transporter. So in the same biotope, Streptomyces tendae secretes 7-CinC, an antibiotic to kill Bacillus subtilis. So Bacillus, in response, overexpresses BMRA to decrease the intracellular concentration of this antibiotic below its cytotoxicity threshold. Moreover, BMRA can expel a variety of substrates. It belongs to multi-drug resistance proteins. So, BMRA are able to transport various and specific ligands using an alternating access mechanism. So let me explain this mechanism. We begin with inward-facing conformation, where the drug can bind to inside of transmembrane domain, and ATP can binds to nucleotide domain to dimerize them that trigger conformational changes within BMRA, which adopt outward-facing conformation, and then the ligand can be released outside of the cell. The next step will be the hydrolysis of ATP to reset the transporter for a new cycle. And this mechanism, this global mechanism, is widely accepted for exporter of type 4, but local details are unknown, and the way to transport all the ligands stay elusive. So my scientific question is to unravel this different state of this mechanism and how ligands can be transported by BMRA. So I use two approaches to answer this question in steady state and time-resolved conditions. And during my talk, I'm going to focus on this transition from inward-facing conformation to outward-facing conformation. We use a trick to get only this forward transition. We use an inactive mutant for ATPase activity. So in steady state condition, we have looked at the binding of ATP. In absence of the drug, there is a Michaelian behavior for the binding of ATP. When there is a drug like rhodamine, you see we get a sigmoidal curve, so there is cooperativity for the binding of ATP. And we are interested to what happens of BMRA with different concentration of ATP, so that we have so many 3D structures of BMRA in detergents to understand this shift. And without going into the details, three main findings. We can visualize the molecular effect of rhodamine because it plays an effector role. And we can visualize the transporter plasticity thanks to molecular dynamics and 3D variability analysis. And other biochemical experiments explain the uncoupling between drug transport and ATPase activity, and this work is currently under Nature Communication. So now, what would happen if we trigger this transition while looking at biochemical and structural level over time? For biochemical part, I can use the tryptophan fluorescence to monitor conformational changes over time. So the experimental setup consists of the addition of the ligand with BMRA. There is a quenching first. So BMRA is the inward-facing conformation with the ligand. When the addition of ATP, we have an increase of fluorescence because the environment of tryptophan are more hydrophobic and less exposed to the rhodamine. Okay, I did many control to understand what I see with time resolved fluorescence. Here's the relevant ones. With buffer, I had buffer. There isn't an increase of fluorescence, so it's not an interfact. And then when I had ATP, instead of ATP, there is nothing. In comparison with ATP, you have the increase of fluorescence. So because the nucleotide domain of BMRA is unable to dimerize with ADP. Also, I can modulate the shift of population with the temperature. The higher the temperature, the quicker the transition done. And I can modulate with ATP concentrations. And these experiments have been realized by our own spectrophotometer. And we aren't accurate in the first seconds. Yes, my dead time is around 3 seconds to add and mix compounds. And for that, that's why we use a stop flow machine in collaboration with Dr. Corey-Milon to get information about initial speed. I won't give you more detail because this work is ongoing, but I have compared the wild type between wild type and mutant. As you can see, the wild type, the transition in the case of wild type is faster than mutant. And I test 2 types of mutation like glutamine or alanine. And as you, we have a difference. This mutation doesn't impact really the binding of ATP, but more likely the conformational changes. Thanks to this technique, I have access to kinetic rate constant and propose a new enzymatic model. And thanks to all these experiments, I identify the best conditions to do time resolved cryo-EM. So the condition with mutants is with 300 micromolar of ATP at 21 degrees. And the idea is to slow down the transition to see all the transition. So in collaboration with Dr. Guichon and Dr. Lefteris Zarkadas, we froze at key times BMRA, and I analyzed the particles of BMRA over time. So, without ATP, we have 100% of inward-facing conformation. At 12 seconds, okay, this time is represents the time to mix the compounds, put on the grids, blot, and freeze. So at 12 seconds, we have a small amount of BMRA in outward-facing conformation. At 12 seconds, we have half EF and half OF. At 40 seconds, the majority of particles is in outward-facing conformation. So over time, the population of outward-facing conformation increases while EF decreases. At the end, we have 100% of outward-facing conformation. And this analysis of this data is still in progress, but we see the perfect match between the trace of fluorescence and the cryo-EM data. To sum up, we can follow population in time resolved cryo-EM, and that matches with biochemical experiments. And the analysis is still ongoing, but icing on the cake, we see transient intermediates, and I'm going to show you an example with 3D variability analysis. So it's a BMRA in inward-facing conformation, and thanks to time resolved Cragonian data, we can see, as you can see, the nucleotide domain of BMRA get closer in response to ATP binding. And the perspective is to understand these local deformations and to elucidate this mechanism in parallel with biochemical experiments. So I'd like to thank the whole team, collaborators, fundings, and thank you for the committee to select my talk and give me the opportunity to present my work. And thank you for your attention.
Thank you.